Good morning, everyone. Welcome to the lecture. Welcome to the lecture on the topic DNA replication in prokaryotes. So today it's time. It's time to read and understand how exactly the replication process goes about. How exactly our cell is able to replicate its own DNA. Now, what we'll be discussing today? See, replication is everybody is aware. The replication we are talking about duplication, the number of DNA or duplicating the DNA. Now, that replication today we'll be focusing only on prokaryotes, although, and in next phase of lecture we'll be focusing on eukaryotes. But today our job will be to understand the replication scenario of prokaryotes because their doubling time is very much lesser than us. Then they must have that effective machinery to replicate the genomic content, replicate their DNA in coherence with their cell division. So what are the players? How exactly that particular application is going about? We had already understood that how replication came into limelight. How it was established that it's a semi-conservative mode of DNA replication. How it was established that yes, the circular double-stranded DNA molecule of a prokaryote is actually forming a replication fork, and that replication fork is moving bidirectionally. It's forming a first a bubble, and that bubble is getting enlarged. So we have established that. After establishing that, we have we will now focus on what are the players of this particular DNA replication, how exactly this replication is going about in different stages, how cell recognizes at what point it has to start the initiation part. After initiation, how the elongation is going on, because we already know that the replication is happening in two forms. The two template strand, the two parent strand, the one strand is going for continuous replication, whether the other is forming the Okazaki fragments, that is semi discontinuous nature. When these are arranged anti-parallelly and the replication can happen only in one direction, how the opposite direction is satisfied, how the need to replicate the lagging strand is satisfied, how those Okazaki fragments are formed. That is our job for today. That is our job for today. So the learning objective of this particular lecture is to give an understanding of how the mechanism of DNA replication is happening and what are the different players that are present in this particular line of event in prokaryotes. So by the end of the lecture, I expect that you should be able to understand that in the stages like initiation, elongation, and termination, how those three stages are being regulated by different proteins. And then at the end, how it is resulting in the formation of double-stranded DNA, a template from a template, a new DNA is formed. So the complete coordination that is required in sequential manner to replicate a DNA, that should be a probable outcome. If you have understood that, then that lecture is a success. Then the lecture is a success. Let us begin. Before directly going, directly putting ourselves, pushing ourselves towards understanding the initiation process. First, you should be able to understand that who is the main player. Who is the main player in that complete process, or who are the main players in those process, whose responsibility is it to take the template DNA and synthesize a new one, which enzyme is responsible. So we already know the enzyme we are calling out is polymerase. What kind of polymerase? See, polymerase is an enzyme. What that enzyme is doing, try to understand how we nomenclate that particular protein. It is using DNA as a template. This already existing DNA strand is utilized to form a new strand. So what it is doing? It is a DNA dependent DNA polymerase. So we are calling it in short form as DNA polymerase, DNA dependent DNA polymerase. So E. coli has three types of DNA polymerase: 1, 2, and 3. All those three have the polymerization capability, but the extent of polymerization and the rate of adding the nucleotides is completely different. So in the table, you can easily compare. The comparative chart is provided to you. You can easily see that the genes which are coding for these enzymes are proteins. So these particular DNA polymerases are also coded by certain enzymes. So the enzymes which are coding this particular scenario or this particular proteins are called as Pol A, Pol B, and Pol C. Pol A, Pol B, and Pol C. These are the genes which are coding these enzymes. Now, DNA polymerase 1 is again a single subunit. Then Pol 2, DNA Pol 2 is 7 subunits, and Pol 3 is more than more than 10 subunits are present. But more importantly, rather than the structural scenario, it's the activity that I'm interested in. See, now all the three enzymes have polymerization activity. They can add nucleotide on the chain and can cause the formation of new DNA from a template DNA. Now, the fastest enzyme or the principal enzyme which is responsible for DNA replication in prokaryote is DNA pol 3. Its rate is adding 250 nucleotides, 2000 nucleotides per second. In a single second, it can form a stretch of 1000 nucleotide, a short stretch of DNA. It's very fast, very efficient, right? Then we have DNA pol 2, which can add at 40 nucleotides per second. Then we have pol 1, which ranges from 16 to 20 nucleotides per second. Then once the DNA is synthesized, see, DNA is being synthesized and the length of the DNA is very high. The length of the DNA is very large. Is there any kind of machinery which is error free? No, absolutely not. The errors can happen. The errors like modified bases, the errors like mismatches, the errors like nucleotide mismatches, it can happen. And our cell or any prokaryotic cell cannot allow those aberrations to move ahead. It cannot allow those damages in the DNA to get incorporated in the future generations. So it has to be repaired. Now, in order to repair that, one should be first able to identify that yes, the damage has occurred. Now, once you have given an examination, you write all your answers. Then what you do at the end, you go through those answers, that whether you have written it properly or not. That is called as proofreading. And similarly, all these three enzymes have proofreading activity. Please understand, DNA replication is going from 5' to 3' direction. The enzyme is moving from 5' to 3'. In order to proofread what it has drawn here, it has to move from 3' to 5' in the opposite direction. And that is called as 3' to 5' exonuclease activity, and that is called as proofreading. All the three polymerase enzymes have this particular activity. And among these three, the pol 1 specifically have another kind of activity called as 5' to 3' exonuclease. 5' to 3' exonuclease. Now, what is 5' to 3'? Simultaneously, as the DNA is being synthesized, it is also being scanned, and that simultaneous scanning is the function of DNA pol 1. And that's why all these three enzymes are very, very essential in prokaryotic system. Although I have mentioned only three, but in actual fact there are 5 different types of enzymes, polymerase enzymes, that are present inside prokaryotes. 5 different enzymes, but we are here focusing on 3 only because we are going for replication and understanding their basic role in replication. So let us talk about the DNA pol 1 since we have started that particular exonuclease activity discussion. I narrated that this particular polymerase 1 has a specific activity which is not present in the other two polymerases, that is 5' to 3', exonuclease activity. That means it can simultaneously scan the particular newly formed DNA whether the damage is there or not. So what it is doing? It is performing a cleanup function during replication, right? It is performing a cleanup function. Whether there is a damage, I'll be going and scanning and going for the repair. So I'm performing a cleanup of a newly formed DNA. Fine. So when this particular 5' to 3' exonuclease activity is going about, then people investigated that why and whether the complete enzyme, what we are calling a DNA pol 1, is able to perform this function, or it's a specific subunit or a catalytic subunit which is present on this enzyme that is performing this function. People have shown that this particular protein, what we are calling it as DNA pol 1, is made up of large subunit and a smaller subunit. The large subunit is called as clean-off fragment. And if we modify this enzyme in such a manner that we remove this small subunit and keep only the larger subunit, still it is able to polymerize it is able to perform its polymerization function, it is able to perform the proofreading activity, it is able to perform the proofreading activity. Fine. Whereas whereas it is not able to perform 5 prime to 3 prime exonuclease activity. That means that this particular unit was responsible for 5 prime to 3 prime activity. So if you don't require that activity, instead of DNA pol 1 in labs, many researchers also use only cleano fragment because it has that particular potential to go for the polymerization, and it has the potential to scan back in the opposite direction and go for the proofreading, and that's all you want when you're working inside the lab. So see, every player, every enzyme has been investigated thoroughly, and their function and structural part has been investigated. So today we are all aware that how replication is going about in prokaryotes, and in what time, at what, by what players, in what manner. So with that scenario, after discussing DNA polymerase, let us go and focus on what are the other players that are involved in this particular reaction. What are the other enzymes? See, apart from DNA polymerase, in prokaryotes itself, there are more than 20 different enzymes and proteins. Enzymes as well as non-enzymatic proteins collectively are involved in completing the process of replication. In completing the process of replication, the entire complex that is resulting in the formation of new DNA, the entire complex of the protein is called as repliosome. Repliosome. Fine. So what are the components of it? See, DNA polymerase that we have already studied. There is a helicase which will be opening up the DNA. It's a double-stranded DNA. Any double-stranded DNA cannot be used for replication. It has to open up, and that opening up is performed by helicase. It is performed by helicase. So once it is opening up, now you please understand there is a circular structure which is double-stranded. Once you have started opening, the remaining strand which is unopened is now tightly wound, and that tightly wound is termed as inducing of supercoiling. Then that torsional stress that is happening on the upstream region has to be released, and that torsional stress is released by topoisomerase, which is also called as gyrase. Topoisomerase, which removes the torsional strain. Then whenever the DNA is being synthesized, it requires a primer, because the polymerase requires a free 3' OH to add a fresh nucleotide. It cannot start the DNA replication from a scratch. Then that primer, the short RNA stretch, is to be synthesized by an enzyme called as DNA primase. Then once this particular double-strand DNA is opened, fine, and the helicase is moving ahead, these are still complementary. If you lose them, if you don't do anything to them, they can again renature, and you will see that helicase has moved ahead, and simultaneously on the back end side, the function what was performed by the helicase in opening is again closed. So you should have certain mechanism to stabilize the single strand, so that they should not again bind to each other, and that function is performed by single strand binding protein. That function is performed by single strand binding proteins. And DNA ligase is responsible for the formation of closing the nicks that are formed, because when the Okazaki fragments will be formed, every time the primer is removed, and then there is sealing of the phosphodiester bond, that particular function is performed by DNA ligase. So out of the complete scenario, I'm just giving you the example of few enzymes that are very crucial in terms of the understanding of DNA replication. But please understand that there are more than 20 different proteins, I just say. The protein may be an enzyme, and it may be a non-enzymatic protein also, which is responsible for carrying out this particular DNA replication in prokaryotes. Now let us start with this particular replication process. Now at the onset, this is divided into 3 categories. We'll be talking about the initiation, how that particular replication process is starting, after starting, how it is replicating, and once the replication is done, how it is terminated. So let us begin with the initiation part. See, in prokaryotes, there is a specific element inside the genome which is responsible for initiating the replication, and we call it as origin of replication, or IC. Origin of replication, or IC, ORIC. Fine. It's a 254 base pair long sequence which is present in the genome. Now, in this particular sequence, there are multiple consensus sequences. By consensus, I mean there are multiple conserved sequences throughout prokaryotes. and those conserved sequence fall in two categories. The two categories I have represented here in terms of colors. What colors? Yellow and blue. Yellow and blue. Now first let us look at this particular yellow stretch. Now this yellow stretch is made up of three consecutive repeats. We call them as tandem repeats. Why? Because they are repeated one after the other. Now the repeat sequence is made up of 13 base pairs, and the sequence is written here: GATC, TNT, TNT, and multiple repeats of T, contributing to a 13 base pair. That means the one box length is 13 base pair, and there are three repeats of 13 base pair, making it a 39 nucleotide long sequence, which is constituted of tandem repeats of number 3. Then there are other domains which have repetitive sequences of the length of 9 base pairs, but those are not tandem. They are present in different positions and different orientation, and those are classified under category of 9 base pair sequence repeats, and the sequence is presented here. Now these two consensus regions are responsible for initiating the DNA replication. Now what is happening? There are two different proteins. One protein is called as DNA A and another is called as DNA B. One is called as DNA A, another is called as DNA B. Now DNA A will come and bind to this particular 9 base pairs repeat sequence, whereas DNA B is specific for binding to these 13 base pair repeats. So what we have established, there is a particular element inside the circular double stranded DNA of prokaryote called as ORIC. In that ORIC, origin of replication, there are two regions. One is tandem repeat of 13 base pair, another one is repeats, short repeats. There are 4 short repeats of 9 base pair, and there are two proteins, DNA A, DNA B, which are specific to them. Then what happens next? How these proteins are going about? How these proteins are going about for this application? Or are these the only proteins, DNA A and DNA B? So no, these are not the only proteins. Minimum of at least 9 different proteins I have listed here for you that are prerequisite for this DNA replication to happen. In nutshell, what they are doing, they are recognizing the site, they are opening the strand, and they have to start the replication. So what is the function? The first protein is called as DNA protein. Now that DNA protein will recognize the origin sequence. The DNA B is actually your helicase. Then there is DNA C protein. We have primase, as discussed. We have single strand binding protein. We have topoisomerase that will be responsible for opening up. Now let us see how that goes about. See, this is your circular double stranded DNA, which is in supercoiled state. The origin of replication is represented in different color with yellow. Let us zoom that particular origin of replication, and we see here that 13 base pair repeats are there and 9 base pair repeats are there, which we had discussed in the previous slide. Now what will happen? Multiple proteins of DNA A will come and bind to these sequence. Now how exactly a single stretch these proteins will bind to this multiple repeats which are present far apart? So what they do, they form a loop, they form a coil in which all the proteins are present, and now they have an easy accessibility of all these 4 repeats. Once the DNA A protein has bind with an expenditure of ATP, now what it does, it has brought this 13 base pair repeat close to these loops, close to these proteins. Now this will signal another protein, which is HU, histone-like proteins. Histone-like protein, not the histones, histone-like proteins. And this recruitment requires again the expenditure of ATP. Now this histone-like protein will be responsible for making a bubble and recruiting DNA C. And the DNA C along with it will bring DNA B, which is actually the helicase, and it will load the helicase DNA B structure on each strand. So you see there are 2 helicases which are open. Now this particular helicase can open up the DNA in this direction. This particular helicase can open up the DNA in right direction. And once the bubble is formed, you see the double strand has been broken up into single strands. Then these single strands are required to be stabilized so that they should not anneal to one another. And that is provided by again single strand binding proteins. and the torsional stress is removed by topoisomerase, which is recruited at the later stages. So what is the chronology? Chronology is, first of all, DNA protein is coming, binding to 9 base repeats in the coiling form. It brings that particular 13 base repeat closer to the coil. Then histone-like proteins are recruited. The histone-like proteins are recruited, they form a bubble. They form a bubble by means they start opening. And then DNA-C protein comes and loads the helicase, that is DNA-B, onto the single strands. And now the helicase is loaded and the initiation is completed. Now the cellular machinery has loaded the helicase. Now the initiation is completed. The signal is already given. Now what is the job? Now the job is to call upon the polymerase. To call upon the polymerase, please come and start performing your function. We have given you an opening window. This is the window where you have to come and start your function. Now the job is elongation. We have started the process. Now please elongate this DNA. Now please go about. Now before elongation, there is a synthesis of primer. Now for the primer, DNA primase needs to be recruited. DNA primer needs to be recruited. Now all these enzymes collectively are forming a bubble. That bubble, what we are representing as replication fork. Replication fork. Now the primase, what we are talking about, is also called as DNA gene, the prokaryotic system, which is a very short nucleotide, 10 to 16 nucleotides are being formed as an RNA primer at the origin. And this RNA primer is utilized for elongation by DNA polymerase 3. So both the strands which are present here are divided into two categories. One is leading, another one is lagging. The leading strand is what on which the newly formed strand will be synthesized continuously, whereas the lagging strand is in which the DNA replication will be happening in a fragment manner and the Okazaki fragments will be formed. Fine. And we had discussed that why these Okazaki fragments are required, because the other strand is in the opposite direction. The replication can only happen in 5' to 3' direction. Fine. So the other strand, which is in the opposite direction, that needs to be synthesized, and that synthesis, that problem, how that problem is resolved, that we'll be focusing on in this particular part. And let us see what exactly I meant by the problem. See, this blue line again I'm representing as a template DNA, 5' to 3' blue, and the opposite is in antiparallel 3' to 5'. When I said the synthesis can only happen in 5' to 3' direction, see, this is a replication fork which is performing the function. Now the new strand which will be formed, it will be formed in the direction of 5' to 3'. And now this is 5' in 3' direction. So you see there is a continuous arrow. The template is utilized and there is a continuous synthesis of this particular DNA. The primer was synthesized here and that is extended from the scratch, right? So this blue line, which is used as a template, is called as leading strand, or this is called as leading strand, which is being synthesized, the new strand. The other opposite strand, now the machinery is moving from right to left as per the PPT, as per the presentation. Now in order to synthesize a strand on the opposite, using the opposite template, it has to move from left to right. Now it is not possible for a machinery to first go from right to left, then again come back to left to right, to and fro motion. That is not possible. Then what should we do? We have to synthesize this also. What should we do? Now this bottom strand is converted into a loop, and loop in a manner that a primer is initiated, and in this stretch where I am highlighting, in this stretch here, the 5' to 3' direction is synthesized continuously, and using this primer the synthesis happens in this direction, whereby the loop, both the strands are moving from right to left. And how that loop is formed, see, this is the plan, 5', it is a template. So in order to synthesize, if you have to synthesize, you have made it into a loop form. Now both of them are in the same direction, and that is exactly achieved inside prokaryotes and eukaryote model. What are the enzymes that are responsible? Helicase is there. We have a primase here, which is responsible for the synthesis of primer. So we understand that at the lagging strand, there will be formation of multiple primers. And how exactly it goes about? This is how exactly it goes about. This is how exactly it goes about. It slides on the DNA. It slides on the DNA, and this slider is nothing but a clamp. This is called as beta sliding clamp. It slides on the template DNA, and this particular clamp is loaded onto the DNA by a clamp loader. Is loaded onto the DNA by a clamp loader. So this particular clamp, what we are showing here, has enzymes of polymerases present on both the arms, and it is responsible for the synthesis in 5' to 3' direction. Now you see it is moving from right to left. It is moving from right to left, and the black arrows are representing the sliding motion of the template DNA, right? But the newly synthesized DNA is in right to left direction. Now, how we solve the lagging strand problem? We have looped it in a manner that the synthesis is happening in same orientation. In same orientation. If the looping was not there, then it was not possible for the bottom strand to have the same orientation. It would have been always in the opposite manner. Now we have looped it to form into a same position, and that is the job of beta clamp and the clamp loader. Beta clamp and the clamp loader. The primase will all keep on synthesizing the primers in the every bubble, the replication fork. Every time when it moves ahead, it forms a new primer, so that the short stretch which is present at the lagging strand can be synthesized, and that short stretch is called as Okazaki fragment. And that short stretch is called as Okazaki fragment. Now once the short stretch is formed and now the clamp is ready to move ahead, what happens? Now what happens? The clamp will be dissociated. This particular beta clamp will get dissociated, and it will release the loop, so that a continuous strand is formed, and it will occupy the strand which is opened up by the helicase, and now it will form a fresh loop, so that the remaining or the other short stretch of Okazaki fragment can be formed. So you see, this is how the complete elongation process is going about. This is how the complete elongation process is going about. How it is going about? It is going about in a manner that once a short stretch is formed here, now I have to release the loop, and I have to incorporate this particular strand which is now needs the replication into this particular zone. So what I'll do, I will remove the clamp. I'll take out this DNA, I'll slide the clamp and put this particular stretch of DNA which needs now replication in this loop. What is happening on the other strand? It is synthesizing continuously. It has no problem. So this is how coordinated efforts are required at every stage. There is a removal of clamp, there is a loading of the clamp, there is a sliding motion, and in this sliding motion it is being oriented in the same manner. And after the same manner, the one strand is continuously synthesizing, other is synthesizing for a short strand every time, and then again the process is repeated multiple times until the complete genomic content is replicated. This is called as elongation. This is called as elongation. And the complete scenario is what I narrated. I have summarized here for you all. When we are talking about the helicase part, let me focus on this. See this helicase. The helicase is DNA B, whereas the primase is called as DNA G. Now these B and G protein forms a constitutive functional unit in every replisome. These two enzymes are called as primosomes. Why? Because one is opening and another is forming a primer. On the leading strand, only one primer is required, whereas on the lagging strand, every time a new primer has to be synthesized. And what is the principal enzyme that is using this primer to extend the DNA? That is DNA pol 3. DNA pol 3 is actually the core polymerase of this particular reaction. It is actually the core polymerase of this particular reaction, which is synthesizing a continuous leading strand, while the other set of DNA pol 3. So see what I meant by the other set. We have two arms here, right? Two arms here. The beta clamp consists of two copies of DNA pol 3. One will be attached here, one will be attached here. The other copy is responsible for synthesis of lagging strand in a looping manner. in a looping manner. And how it forms a loop, that's what we have discussed here. So when synthesis of an Okazaki fragment is duly completed, the replication fork stops for a while. It The loop which is synthesized already is removed from the clamp. The clamp is dissociated, the loop is removed, the loop is taken ahead, and the ahead portion which was opened up by the helicase is now taken into loop, so that the other remaining portion can be synthesized and the new clamp is formed. So every time the process is repeated and the new Okazaki fragment is formed. So we saw that in elongation, it is a requirement of multiple proteins that are present here, the multiple proteins. So when we talk about the repliosome in terms of the rate of forming the DNA, rate of replication, we established that it is forming at the rate of 1000 nucleotides per second when we are talking about the DNA poly 3. When we are talking about the DNA poly 3, once the replication has taken place, then you must understand that at the lagging strand, the short fragments are synthesized, and every fragment needs to be joined, and this RNA primer needs to be removed, because you cannot incorporate primers, you cannot incorporate an RNA molecule in between. Then who performs this function? The DNA polymerase 1. The DNA polymerase will come, it will see that there is a gap and there's a primer, I need to remove this primer, will be removed, and the polymerase 1 will come and extend this particular arm, and the final sealing will be performed by DNA ligase, Now DNA ligase, every time when it has to seal that particular gap, requires an ATP and an additional cofactor of NAD. Now, if these cofactors are present, the energy expenditure is there, your complete lagging strand will be now continuous. Then the last step is your termination. How exactly it is terminated? See, it's a circular molecule. Look at this picture. If I'm starting here and my replication fork, one fork is moving counterclockwise and the other is moving counterclockwise. Eventually, if nothing happens, these two forks will be meeting in the opposite direction, one from counterclockwise, other one from the clockwise. And once they meet on the opposite direction, the replication fork dissociates. And the termination happens eventually. But apart from this, also there are certain specialized proteins that are responsible for terminating the sequence, terminating the DNA replication process. In E. coli, if we are talking about, there are multiple sequences which are called as TER sequences. This pink color represents TER sequences. These TER sequences function as binding sites for TUS protein. What is TUS protein? Terminus utilization substance. The TUS protein will be binding to this particular site, and it will halt the replication fork, and this halting in the replication fork will be resulting in the dissociation of the complete repliosome. Now there are multiple sites of TER gene sequence. Not all the sites are active always in the replication. Out of this, only one site anytime will be active, and at that particular site the termination will be happening. The counterclockwise fork will be coming, suppose at the B, the clockwise came at the B, and they both dissociated. The remaining sequence, which is actually the TER B sequence, the remaining stretch of short 100 to 200 nucleotides, how it is replicated, that is still under investigation, that how these are actually replicated, and the complete sealing is going about. So you see, when either of the replication fork is encountering, it can dissociate itself, and the functional form of TER sequence binded to a TUS protein out of that complex will encounter the replication fork and cause the termination and dissociation of that particular part. Then the remaining portion where exactly the TER sequence is present, how those few hundred base pairs we are talking about are getting replicated, that is still in investigation, and multiple mechanisms have been showcased in the literature. So what we have done till now, what we have studied till now, we have completely established that how these processes are going about, in terms of initiation, in terms of elongation, in terms of termination. How DNA A, B, C proteins initiated the site of replication at origin of replication ori C. How those repeats, tandem repeats and 9 base pair repeats plays a crucial role. And then elongation wise, how that beta clamp is involved in this particular replication, how that clamp loader is performing its function, how the lagging strand is forming a loop so that the anti-parallel direction can be converted into a parallel direction. but the direction is always 5 prime to 3 prime. Then after that, how the sliding of DNA is happening? How the sliding of DNA is happening? Then once the sliding has happened, what is the function of primase? That every time it has to synthesize a primer when the Okazaki fragments are being synthesized. Once the complete replication fork has gone for elongation, the DNA polymerase 1 and ligase has removed, polymerase has removed the primer, the synthesis has taken place, the last bonds are being sealed, then the termination has to happen. Then the termination is eventually mediated by the ter and ter sequences inside a prokaryote, or we should say E. coli, specifically E. coli. Out of those termination sequence, only one sequence is activated, and that activated sequence, once it the replication fork is bidirectional in prokaryote, right? So then when they're moving in bidirectional, then eventually it will move to the opposite end, and eventually it will collide and will dissociate. So you see, that is how the termination process is going about, and that is in turn the that is in all the summary what one should be able to know when we are talking about DNA replication, specifically in prokaryote. In eukaryotes, if you see, there will be many more proteins, many more complex structures, but there will be analogous to these particular prokaryotic proteins. So based on this, let us go through the MCQs that are very much important when we want to summarize a given topic. Then DNA replication always proceeds in what direction? Always proceeds in what direction? 5' to 3' direction, because you cannot go in opposite direction. That's why the lagging strand is looping out. The replication of lagging strand occurs in the form of what fragments? The incoming nucleotide. When the incoming nucleotide has to bind to a already existing nucleotide, what bond is being formed? The phosphodiester bond is formed between what? How many DNA polymerases are present here? I told you how many DNA polymerases are present here, that you can answer easily, although we have discussed only 1 to 3. The proofreading activity, what is proofreading activity? That also we have established, that one has to scan the DNA while the process is being going about. Then whether it is semi-conservative, it is conservative, or it is an isolated process. And who exactly established this, that it's a semi-conservative process? Was it Michelson and Stahl? Was it Watson and Crick who actually gave the structure of DNA? Or it was John Keynes who actually worked on the prokaryotic model, who actually defined that yes, the replication fork is there, who actually coined the term of replication fork? Now, who proved that DNA replication can occur simultaneously on both the strands? It is not happening on the other strand and it's not happening on the other strand. Out of these four, that remains to be answered. That is your job after reading this. If you have the curiosity to check who did this, then you should be able to get it. And in the Michelson style experiment, how exactly they decipher that? How exactly they decipher the nature what they're talking about? What kind of isotope they use? And in John Carrian's, how he worked on this. So these two aspects, what we had discussed also in that manner, that Michelson style and John Carrian's, what functions they perform, that is very, very much important, that you can find out in the literature. And as per your curiosity, you can read those particular topics. So in nutshell, I have also summarized certain references which you should read in order to have a much better understanding of this DNA replication in prokaryotes. And there are certain books, there are certain NPTEL lectures, and of course the research articles or the review articles which completely and beautifully summarizes all the aspects of DNA replication. Then one should always read that. So friends, we will stop here and hope this lecture was fruitful to you. We'll meet in another lecture. Till then, please stay safe. Thank you.